Hello everyone. So today let's discuss the topic endosperm formation. Okay, that is the next part of the post-fertilization events. Earlier we have studied the first process of post-fertilization event, that is the embryo development, and this is the second part, that is the endosperm development. Now, as already discussed earlier, endosperm development takes place when the male gamete fuses with the central cell or the definitive nucleus to form a triploid structure. Okay, that is triple fusion takes place and forms a triploid structure which is called the endosperm. Now, endosperm, it is actually a nutritive tissue which is meant for nourishing the embryo in the seed plants. That means it is a food laden tissue and it will help in providing nutrition or food to the developing embryo. Now, in the female gametophyte, this tissue is present, okay, that is within the embryo sac. Now, in angiosperm, the endosperm is actually a tissue, special type of tissue which is formed as a result of vegetative fertilization or triple fusion or fusion of the male and the diploid secondary nucleus. Okay. Now, this fusion product is the primary endosperm cell having a triploid endosperm nucleus. Now the development of endosperm, it basically takes place by three processes. Okay, that means during the development we get three different types of endosperms. So first is the nuclear type. So this is the nuclear type of endosperm, as you can see in the diagram itself. Here it is the common type of endosperm, okay, where the primary endosperm nucleus, it divides and redivides to form a large number of free nuclei. Now, as you can see, this is the embryo sac, this is the developing embryo, and here the endosperm cells are repeatedly dividing, okay, and they are forming a large number of free nuclei, thus giving rise to a multinucleate endosperm. So this is multinucleate because this is one particular tissue, isn't it? A central vacuole appears gradually. As you can see here, this is the central vacuole. It appears and pushes the cytoplasm towards the periphery. Okay, so it is pushing it towards the periphery. The cytoplasm gradually thickens so that the vacuole decreases in size and ultimately disappears. Okay, that means whole thing is completely filled up, as is you can see here. The wall formation or cytokinesis begins from the periphery. That means from the sides it will start developing the walls towards the center. It makes the endosperm a multicellular tissue. Now, as you can see, this is a multicellular tissue, isn't it? Now, in some cases what happened, the cell wall formation remains incomplete. For example, coconut, okay, which has multicellular endosperm in the outer part and free nuclear or vacuolated endosperm in the center, that is the coconut milk. It is a liquid endosperm, okay. The coconut milk, which is highly nutritious energy drink and it is rich in minerals and vitamins, okay, this is considered as the liquid endosperm. So this is a very simple process, okay. Initially this zygote, it will develop the embryo, it will gradually development start here, and here the cells will first divide, nucleus divides, okay, it forms a multinucleate structure. This is the embryo sac when large number of nuclei are present. Gradually central vacuole occurs, for which the cells nucleus are sent towards the periphery. Gradually the cytoplasm thickens and cell wall formation starts from the periphery towards the center, and as a result what happened, the central vacuole gradually disappears, okay. The second type of endosperm is the cellular type, okay. Here division of the primary endosperm nucleus is followed by cytokinesis, sorry. That means as soon as the nucleus divides, immediately wall formation takes place. That means cytokinesis take place and wall formation take place, and the endosperm does become cellular from the very beginning. Here only during the last part it is becoming cellular, but here initially from the very first stage it is becoming cellular. That's why it is known as cellular endosperm. The third is the halobial endosperm, which is an intermediate type between the cellular and the nuclear endosperm, okay. Now it occurs in maximum monocots. Okay, the first division of the primary endosperm nucleus is followed by a transverse cytokinesis. That means this is the first division which will take place. Okay, thus two unequal cells are formed, the larger micropylar and smaller chalagel. Okay, micropylar, it means it is consisting of the embryo. So this is the larger one, this is the smaller one. The further development in both the cells occur like that of the nucleus. So this is the first cell. First division, wall formation take place. Then next time what happen, nuclear type of endosperm development starts. That means the nucleus of each part will divide to form a multinucleate structure. Then gradually a central vacuole will is created, and this central vacuole pushes the nucleus and the cytoplasm towards the periphery. Then gradually the cytoplasm thickens and makes the vacuole smaller, and gradually here cell wall formation or development starts, and finally it develops into a cellular type. So in this type of endosperm development we see both the types, that is cellular as well as nuclear type. Okay, now here, as you can see in the diagram itself, the chamber towards the chalagel end, it always remains smaller, and later on what happened, this degenerate because the embryo is present in this larger part. So definitely this is the most important part. Okay, so this one later degenerates. Okay, so these are the three types of endosperm development processes. Very simple. A nuclear type, okay, where first nuclei are forming, later cell wall formation starts. Cellular type, in which from the very beginning cell wall formation goes on. Halobial type, where first cell wall formation then follows nuclear type of endosperm development. That means it is an intermediate step between the two types of in endosperm development. Okay, now the endosperm, it is actually meant for nourishing the embryo. Now during its growth actually it crushes the nucellus. Okay, that means when the embryo is absorbing the nutrition, that means it will definitely crush the cells. Now here only you can see when this is developing, definitely it will crush the cells and absorb the nutrients. Now in dicots, the food reserve present in the endosperm is completely consumed by the growing embryo. Okay, and then that particular type of seeds which are developed are called the non-endospermic or ex-albuminous seeds. Okay, then in some plants like bean, pea, etc., here the reserve food is stored for future use of the embryo. Then that is called and the seedling is kept in either nucellus or the cotyledon. Okay, then in cereals, coconut and some plants, the endosperm enlarges and retain the reserve food in the mature seeds. Then those are known as the endospermic seeds. Now in some like phoenix, erica, here the endosperm is quite hard. Okay, and the endosperm, it becomes convoluted in some plants like passiflora, and it is then known as the ruminate endosperm. Okay, so these are some important terms related to the endosperm development, but basically when you are studying about the development, you have to properly mention nuclear type, cellular type and the halobial type. Next, after endosperm, the next thing what we get is the perisperm. So what is actually perisperm? It is the unused persistent residual nucellus left in the seed outside the endosperm. That means when the embryo is developing, outside the nucellus is definitely present, isn't it? Sometimes what happened, that particular nucellus is left out. So that is called the perisperm. It is one of the food storing tissues of the seed. Okay, and it originates from the nucellus and it is purely maternal type. Okay, the cells are all diploid and are found surrounding the endosperm. The nutrition of the perisperm is completely absorbed by the endosperm. Okay, that means it is actually the nucellus part, isn't it? So that particular nucellus is completely absorbed by the endosperm tissue. Now it is basically found in families like Chenopodiaceae, Peperaceae, okay, like black pepper, sugar beet, etc. Now after this, let us study the structure of seed. Okay, so seed, that is the final product of the sexual reproduction, or we can call it as the ripened ovule. Okay, now this particular structure, it actually develops inside the fruit. It consists of one or two coats, okay, seed coats, one or two cotyledons. If monocot, one cotyledon; if dicot, two cotyledon, and an embryo axis. The stored food for the use of the embryo, so that the embryo can later use it, is present either in the cotyledon, okay, or in the endosperm. And when such type of seeds which is storing the endosperm, okay, or the food is stored in the form of endosperm, then those particular seeds are known as the non-endospermic or ex-albuminous seeds, as for example pea, gram, etc. Now in albuminous or endospermic seed, the whole endosperm is not consumed during the development of the embryo, but a sufficient amount is stored for the future use. For example, wheat, maize, etc. Again, in some seeds, the residual nucellus persists and it is known as the perisperm, okay, as already we have discussed just now. Then when the embryo reaches maturity, then it further grows, and that particular growth is suspended due to the development of some growth inhibitors, abscission of funiculus, and changes in the integument. Okay, so these are the different types of things that are related to the seeds, that is the ultimate product of the sexual reproduction. Now when the seed grows, that means when the ovule ripens, the walls of cells of the integument, they lose their protoplasm and becomes thick and impermeable, okay, and the integuments that does, it gets transformed into the seed coat. Now as you can see here, this is the integument, so they are forming the seed coat. Okay, the moisture content of the seed falls down and reaches about 10 to 15%. The micropyle present in the ovule persists in the seed as a small pore. Now as you can see, this is the micropyle. Okay, it is meant for passage of oxygen and water in germinating seed. That means when the seed absorbs water, it will germinate, and that germ tube comes out through the micropylar end. Okay, the funiculus, abscissus, and the hilum appears as a scar over the surface of the seed. Okay, so this particular structure, this is just a scar, that is the hilum. The embryo enters a state of inactivity or dormancy. That means when the seed is fully matured, the embryo will remain there. Its activity remains suspended within the seed, and it germinates only under favorable condition. That means the seed, this is the layers of seed, okay, the testa and the tegmen. So these will ultimately protect the embryo within its structure so that it can germinate and develop during its favorable condition. Now as you can see, this is a bean seed. Okay, if we are just looking through the different structures, it is an open seed. Now this is the plumule, shoot apex, okay, this is the radical, root apex, root initiation, and these are the cotyledons. This is the outer layer, that is the testa. If we see a castor seed, then this is the hilum portion, this is the raphe, okay, then this is the thalagel end, this further develops, okay, this is the testa, this is the endosperm, these are cotyledons, and finally this is the ultimate structure. This is the plumule from which the shoot will develop, okay, this is tegment, inner layer is called tegment, outer seed coat is called testa. Got it? This is the root axis where hypocotyl, from which the radicals will start developing. Again, this is a grain of maize or a maize seed, okay, it's a monocot, so its structure is a bit different. Okay, now here aleuron layers are present. This is the testa, endosperm tissue, this is the developing embryo. Here coleoptile is present, the sheath of the plumule, scutellum, the cotyledon, okay, plumule, this is the shoot initiation, embryo axis is present, radicle is there, and coleorhiza is present, that is the sheath covering the root cap. So these are the different types of seeds, okay, which are observed in different types of plants. Now basically the main structure remains same. The outer coats, the integuments, which hardens to form the seed coat. And the seed coat, the outer layer is known as testa and the inner layer is known as tegment, isn't it? These basic things are similar. Then within the testa and tegment encloses the embryo, which develops plumule, radicle and cotyledon, from which later the plant develops. It germinates through the micropylar end, it absorbs water and moisture, it germinates and put forth the seedling. Now when we are discussing about the viability, that means it is the period of time for which the seed retains the ability to germinate. Okay, now already we are telling that within the seed coat the embryo remains dormant. Okay, that means it is preserved for the further approaching favorable conditions. So till how much it can survive there, it can remain there, and when germination takes place during favorable condition, that time period is known as the viability. Now the viability, it actually determines genetically and environmentally. Okay, the viability, it may range from few weeks to several years. It depends upon different types of plants, and basically it can be known by two different methods. Okay, one is how the seed is germinating, and by next is the ability to respire. We can test them and confirm their viability. Now seeds are important because seed formation is connected with pollination and fertilization, that is independent of water. Now the seed, it is an evolutionary achievement. It provides protection to embryo, which is the most delicate stage in the life history of a plant, because embryo is the only thing from which the later plant will develop, isn't it? So it is providing full protection to the developing embryo. The seed is dry with dormant embryo and thick protective seed coat. It is the most suitable and perennation through unfavorable periods. They have adaptive strategies to get dispersed to new habitats and colonize. Okay. Then as the seeds are formed through sexual reproduction, they carry a number of variations. Then these are the major source of human diet. Now many seeds are there which we use for our food, isn't it? Like the oil seeds which yield oil, then spices, beverages, isn't it? And they are the basis of agriculture. So these are the main important things regarding the seed. So hope this video is clear. Go through it properly, go through the different types of seeds, the structure, okay, and if you have any problem then let me know.